Hello, everyone. My name is Franko Piskulich. I have been working under the Dr. Daniel O'Connor lab for the past 4 semesters under the supervision of Dr. Varun Chotsky. And today I will be presenting my project, which is titled Mapping Electrode Placement and Channel Rhodopsin Expression in the Mouse Locus Coeruleus. So before I start with my project, um, Before I start with my project, I will be talking about what is this locus coeruleus. Just how I was 4 semesters ago, I did not know what the locus coeruleus was, but the locus coeruleus is a small, important nucleus located in the brainstem and serves as a primary source of norepinephrine production. For simplicity's sake, I will be mentioning the locus coeruleus as the LC, but as you can see on this figure, it is a very small blue, also known as the blue spot, but the LC has widespread projections throughout the brain to regions including the cortex, hippocampus, and the cerebellum that influence various psychological and cognitive functions. These projections include norepinephrine projections that modulate arousal states and adaptive behaviors, additionally playing a role in regulating functions such as attention, memory, stress responses, and emotional processing. It has been noted that dysfunction of the LC system has been implicated in neurological and neuropsychiatric disorders such as Parkinson's disease, Alzheimer's disease, depression, and anxiety. So historically, the locus coeruleus was viewed as a functionally uniform nucleus or a central broadcasting hub. The idea that its noradrenergic neurons fired in synchrony, releasing norepinephrine widely across the brain to coordinate global behavioral states like arousal and stress. This model assumed broad, nonspecific projections and homogeneous function, as shown in the figure right here, a uniform blue color. But recent work has completely shifted the view. Um, Thanks to advances in viral tracing, optogenetics, and single cell recordings, we know that LC neurons are far more diverse than we thought. Many locus coeruleus neurons have more selective projection patterns, targeting specific brain regions like the hippocampus, etc. In the schematic, each color represents a population of locus coeruleus neurons that project strongly to a preferred field. For example, the frontal cortex with medium green. Note that the schematic is only illustrative and that there are far more than 6 LC pathways. Subsets of locus coeruleus neurons form modular circuits that can influence functions. This modularity means that locus coeruleus neurons don't always fire together. Instead, they form subpopulations that activate based on behavioral context. This is where single unit electrophysiology comes in. By recording from individual LC neurons, we can now observe heterogeneous firing patterns that correlate with specific task or environmental cues. However, two major technical challenges exist. One, the LC is very small. In rodents, it's just alone around 3,000 neurons. In addition, it's very deep into the brainstem, making electro-targeting difficult. Second, it's difficult to be sure you're recording from the LC neurons without any additional identification. That's where optogenetics, in particular optotagging, has become essential. In our study, we express channel rhodopsin in LC neurons using a mouse line, which is DBH create crossed with AI32 mouse lines. By delivering blue light to the LC, we can identify neurons that respond with increased firing during stimulation. This gives us this gives us confidence that the action potentials we are recording are from the LC neurons. This figure illustrates the pathway of norepinephrine within the noradrenergic neuron, such as those found in the LC. This process begins with the amino acid tyrosine, which is converted into L-DOPA by the enzyme tyrosine hydroxylase, or TH. This is a critical step because TH is the rate-limiting enzyme as well as a key molecular marker used in our study. L-DOPA is then converted into dopamine by the enzyme dopa decarbylase, Dopamine is then transported into synaptic vesicles where it is further converted into norepinephrine by dopamine B hydroxylase. Norepinephrine is then released into the synaptic cleft and acts on various adiponeric receptors. The presence of TH was used to confirm the identity of locus coeruleus neurons expressing channel rhodopsin, ensuring that optogenetically responsive cells were truly noradrenergic. So then this gets to my project, in which we then are identifying the co-localization of C channel rhodopsin, GFP expression, and tyrosine hydroxylase, confirming LC neurons, as well as locating the electrode tracks that align with the locus coeruleus region for accurate and reliable recordings. The methods in our study, I will run through this really quickly, but essentially to analyze the animal's brains and the lesion's location after recording, after our behavioral recordings, we slice the brain into thin layers, which were 100 microns thick. And to do that, first, the mouse, after its recordings were perfused and and the brains were extracted, then we would slice the brains using a slicer, 100 microns thick, and then the animal brain sections were immunostained with antibodies that identified the LC neurons, and then eventually mounted on slides and imaged using three various lights to analyze DAPI, GFP, and TEH. Here's an atlas to show where the locus coeruleus is very prevalent. As shown on the left, LC is more prominent when the cerebellum and the inferior colliculus meet one another at negative 5.52 millimeters posterior of the bregma. The left, and then on the right, sorry, the most anterior part of the LC will disappear once the cerebellum has been fully sectioned and no longer surrounds the inferior colliculus. And that's around negative 5.34 from posterior of the bregma. So this just shows that the LC is very small. It's only about a 200 micron distance from these two images that is the center of the locus coeruleus. So then that transitions us um, to our images in which we were able to confirm the expression of locus coeruleus neurons. So here are one of our mouse sections, and these are all coronal sections at negative 0.32 posterior from the bregma. So around this picture, this slice right here, or this location. And as you can see here, from the TH expression, GFP expression, and our composite, that the dual labeling successfully confirms the expression of channel rhodopsin, at least for the GFP, which indicates that these are locus coeruleus neurons, and this is the region of the locus coeruleus. So how does that play into our electrode placement? We want our electrode tracks to be targeted around this area. So previously, when I started the lab, one of our earlier mouse subjects, our electrodes were not very accurate. As you can see here in our left image, this image is a very posterior slice. So looking at our atlas around this area right here, approximately negative 5.6 posterior from the bregma. And as you can see here, there's damaged tissue. This damaged tissue suggests that it was from the targeting of the electrode. However, if you look at the image on the right, this is exactly at negative 5.32 posterior from the bregma, so around this area right here. But there is no tissue damage, so therefore we can assume that the electrode did not target the area we specifically wanted for the locus coeruleus. And you can see as well on the based off the the biomarker expression is not very prominent as compared to where we know the locus coeruleus is located. Here is another example of another mouse, earlier mouse that we sectioned, we stained and sectioned. There is a lesion marking at um, negative 5.4 posterior from the bregma, but there isn't any markings at the region where we know the locus coeruleus is at, or the region of the dual expression of the biomarker, as compared to our more posterior slide. As you can see, there are significant tissue damage where the locus coeruleus should have been if the electrode was placed 250 microns anterior. So we know from this two mice, we're able to ensure that for future animals and future behavioral recordings, that the electrode should be adjusted anteriorly about 250 microns, and that will then target the region of the LC that is very prominent. So here is our later mouse lines that we used, that we sectioned. So based off our improvements, as you can see here with the pointer, we have an electrode lesion at negative 5.5 posterior from the bregma. And we have these lesion marks that are indicated of the electrode at our LC center region. And you can see on the right, there are no lesions present because this is a very posterior slide. So this slide is at negative 5.6 from posterior from the bregma, which was around this slide right here. And before, we used to target the electrode at this area, which was way too posterior of the locus coeruleus, and we adjusted our electrode so that we won't have the same mistake. And because of our changes, we have led to a successful recording of 8 different mice. So this figure shows the raw voltage tracings recorded from the single electrode implanted in the brainstem and from our accurate recordings. The black waveform represents the electroactivity picked up by the electrode over time, and each orange dot corresponds to the action potential. The light bars represent periods when blue light was delivered through an optical fiber, and when blue light is on, it depolarizes the channel rhodopsin expressing neurons, causing them to fire. We can see that the firing rate increases dramatically during these light pulses. The spike pattern provides strong evidence that the recorded neuron is responsive to light and is likely a channel rhodopsin expressing LC neuron. And on the right, we have is the action potential that confirms that we did isolate a single neuron. Together, these plots validate both the specificity of channel rhodopsin expression and as well the precision of our electrode placement in targeting locus coeruleus neurons. This project contributes to future research that locus coeruleus neurons do not act as a uniform broadcast system, but rather as distinct modules projecting to specific brain regions. Looking forward, this project opens a door for more targeted behavioral experiments where researchers can map the impact of defined locus coeruleus neuron subpopulations on learning and adaptation. As we refine our tools for manipulating and recording for these circuits, we can better link neurodynamics to behavior with clarity. Ultimately, this project validates a framework for locus coeruleus targeting using electrodes. Thank you, and for any direct questions or comments, please feel free to email me. And special thank you to the O'Connor Lab and my mentor, Dr. Varun Chotsky. Thank you.